today we're going to be talking about plant propagation. So this talk, I will take notes on and use examples of California native plants, but this is just an introduction to kind of teach you. There's a lot of science involved with doing propagation. You got to really know what the plant is. It's more than just putting it in the ground and garden and watering and feeding it in the garden setting for those types of blooms. This you really kind of need to know the anatomy of plants. You need to know kind of what type of plants they are to be able to reproduce them or create new individuals, which is that propagation. So again, if anybody came in a little late, we do have handouts. I have all the notes here available for you, so you guys can just listen and really try to understand and get these concepts down. But if you don't want to copy or if you want to save some paper, there is this QR code if you guys want to scan that, and all the notes on PDF will come up for you. Okay, so a little bit of an overview of this talk. We need to define some of our terms. So a lot of people will kind of misuse some of these terms, but if you really understand them as far as biology and botany and propagation, horticulture, what does this all mean specifically? It really helps you understand what you're doing if you're trying to grow plants. Then I'm going to go over some of the basics and the different traditional methods of growing native plants or plants in general. And then also go into some advanced methods. So there's the traditional methods of growing from seeds or cuttings. That's what people are more commonly familiar with. But there are some more advanced methods when those are more challenging. So things like growing ferns from spores or air layering, and I'll get into all these topics, and then something that I've been really working on lately, which is plant tissue culture, and I'll get into what the specifics are on that. So what is biology? It's kind of a real umbrella term, and it encompasses a lot of these other types of things. So biology is the study of life. So that includes medical professionals, those are biologists. It includes wildlife and people out in the field studying animals. That's wildlife biology. But a section of that study of life is the study of plant life, and that is botany. So botany, kind of in a working term, is more for, it goes a lot towards taxonomy. More traditionally is how people use botany and which plants are related to what, and that whole kind of tree of life is used towards botany. But really, it's just the study of plants. And so from that is also horticulture. So horticulture is not really botany per se, but it's a specific kind of practical application of plants. And so what that term means is it really is just taking care of a plant or bringing it into your garden. So hortus or garden and cultura or cultus means to cultivate. So if you have a garden and you're cultivating plants and you're growing plants in your garden and taking care of them, that's horticulture. And so when you're out there pruning and fertilizing and choosing the right plant and its design and where you want it, that's horticulture. But plant propagation is a little different. Plant propagation is producing new plants, or the production. So my job title here at Tree of Life, I am the production manager. So my job is to make sure that we're growing plants. And new plants, not just growing them like in a garden, but growing them in the sense that I'm creating new plants and having these available for everybody to purchase, to bring in their home so that they can do horticulture. Unless you want to do propagation too, which I'll teach you some basics in this talk, but I'll move on from there. So producing plants, that's either done two different ways in a grander scheme. So it's either done sexually, and it means an exchange of genetics, exchange of genes, just like how we all do when we have our offspring. The same thing with plants. But you can also do that asexually, which is like a clone. We're not quite there yet with us, but we are with plants. And plants lend themselves pretty well to this. In fact, some in the wild do this naturally. And so that's how we can take advantage of what these plants are doing to be able to grow new plants. So the sexual reproduction, that's all done with seeds. So if you're getting seeds, you're getting an exchange of genetics and that genetic mixing, and you can have different variations of plants. much like I am very different from my brother, but we are still very closely related. And so that's where the sexual reproduction is. And then asexually is what you would see as a clone if you were to hear someone say, I want to take a cutting of that. And so if you take a cutting, it's literally a cutting, and then it roots, and the plants have this ability to create a brand new plant from that. And that's a genetically identical plant. It's the same plant in some aspects, but you can have multiple individuals. And that's how you can do, or that's asexual replacement, or without sex is what that means. And that's a clone. Now, grasses and other plants like this, so this is a festuca, so it's a fescue, and I believe this one is one called Patrick's Point, no, Siskiyou Blue. And so this has a certain trait, a certain color, a certain growth habitat. And so I don't want to grow this from seed, or else I would get the different variation. I mean, if it didn't matter, I would definitely grow some from seed, but if I want that exact trait, or that one that we put a name on and said that's Siskiyou Blue, it's got an excellent blue color, then I want to clone this. I want the exact plant, the exact traits and everything. So that's where you would take a clone and do asexual propagation. Now, there's a term called division that you do with grasses. So this has a whole bunch of clumps and roots, and there's little rhizomes together, and they're all bunched together. You can divide that, break it up, and have new individuals that way. That's also asexual reproduction. It's kind of like a cutting, but the anatomy of the plant is different, and so that's called division. So then we'll get into seeds. Seeds are incredible things. Nature figured out an insane science to be able to reproduce new individuals in the wild and to continue plants into the future and everything. Seed science, it blows my mind. So there's different methods, and this is more towards ecology of plants, is that the way in which they disperse. These are their strategies to get their propagules, so propagation, to grow new individuals. So propagules are seeds, cuttings, the starting material for propagation, or to create a new plant. So the method in which these plants do it, so like the poppies we have here, our state flower, you may have seen this in the garden, hence the name. I'm assuming that poppy comes from this type of method in which it gets its seeds out, is it pops, it throws its seeds out. You'll see this in a few other plants. So there's plants like this Ruellia down in Baja. When a monsoonal rain comes, or I think Caesalpinias, or these desert kind of bird of paradises, as soon as their seed pods get this kind of warm temperature and they're nice and ripe, and a monsoonal rain comes, it triggers that plant, hey, we're going to have some moisture here, and it pops the seeds and explodes. You can see this in the garden. Sometimes we've watered them over here, and as soon as we come in on a nice hot day and we're watering with our hose and we hit it, the seed just disperses everywhere. Now, why does it do that and not just drop its seed? It's because, one, there's going to be competition with the plants and resources and things there, but it's also trying to get the seed out and into other areas and expand on its habitat, and expand on its distribution, and have different individuals more out in the wild. Another one is wind. Has anyone grown milkweed? Milkweeds are very, very popular now, right? So the host plant for the monarch butterfly. But if you see the seed pods on them, they'll have all these kind of silk-like plumes on them, on each seed. And what that does is it's so light, and the seed's pretty light, that it can actually carry and travel towards other places with using the energy of the wind to be able to distribute it. Because these plants are stuck in the ground, right? They're not walking up and going and, finding new territory. They have to have a method of doing that, and they use the energy of different things physically to get the seed other places. Actually, this cottonwood over here, if anyone's ever seen the cottonwood in the summer, or when it just starts to, right in spring and fall, when it starts to just have all these raining down of the seed, that's taking advantage of that wind with that same type of method of having silk and things to move and transport that seed. Another one is floating. So bladder pod is an example of that. You have these nice big air bladders on this plant, and so those can move pretty well through downstream, or even in the wind. It'll start to use that kind of bag to move it in other places. But the most iconic of that is coconuts. So these are big seeds that are real able to float and mix and use currents to go towards different islands or wherever that might be along the coast. And so that's another, it's using the physical traits of water and things to move that seed. Because that coconut is a seed. It's just a very big seed. And we'll get more into that. And then animals. This one's interesting. So when a bird eats a seed, or a coyote or a fox, the island fox I've seen out on the islands, they love toyon berries and a lot of different other things, and they're eating that berry. It's digesting it, and it's using all that kind of stomach acid to scarcify it, and then it expels that, you know how, and then that now lives and distributes and uses the energy of the animal to transport their offspring. All right, so when I think of seeds, I'm a big comic book nerd, and this is when I kind of saw how this was a kind of a metaphor with Superman. If you guys aren't familiar with Superman, I'll give you a little brief synopsis. So I look at seeds as little transport vessels, and there's a little baby inside. And so you can see on this diagram over here, you have a protective coating, you have all this food and sugars and fats or carbohydrates within the plant to help and feed that little baby. But there is a little baby. You see the little baby plant with the leaves and everything on there. And so it's there, but it needs to get out and transport. So for the example of Superman, so on the planet Krypton was going to explode, and Kal-El and his parents wanted to have hope for the future. And so they took their only son and they put him in a vessel and sent him out to Earth. And so they put him in that transport vessel. And I see that kind of similar with seeds. You have maybe a different changing environment, so climate change or just natural different changes in the seasons. That's what the plant is doing. I need to get seed out. I need to get my offspring out so we as a species can survive. And so once it finds a nice new habitat, all the conditions are right, you're going to have that seed to start to germinate. And so it's going to use the energy from that transport pod that was packed a lunch, that the mom packs a lunch. My mom always packed a lunch. And then a root will then set down. It'll then draw up and use the nutrients and water from that new environment, and then be able to create its own new cells and be able to put on new leaves and everything and use the power of the sun, much like Superman did, and he became new and bigger and stronger than he ever did. That's how I kind of see plants. So when you grow plants, there's a lot in that seed. Sometimes there's a hardened shell and other chaff and material in and around the plant. So when we grow plants here for propagation, to grow plants in a unique setting, this is not naturally, we have to clean that seed. And so, because if you don't, all that extra material can harbor fungus and other things that we'll just want to eat and break down and do what they do naturally, which is break down material in the environment. And so it can do that with seed. And seed, again, has a lot of carbohydrates and energy stored in there. And that's what almost anything really wants. That's what we do when we eat food. We want that kind of energy. And so we need to clean it. So we use screens, we dehydrate seed or fruits, and then mash those and clean all that out to be able to then grow them and produce it. So if you're going to do propagation, you're going to look at different methods on how to clean that seed and get to the actual seed to start new plants. So seeds have different ways that they actually inhibit themselves from germinating. It's kind of counterintuitive, right? You'd think a plant or a seed would want to do everything it can to germinate and grow. But you only got one shot at that, right? And it's such a vulnerable little thing, this little seedling. So nature and these species have evolved to have different ways of stopping germination until the right conditions are present. So there's hormones within the seed that stops and makes sure that doesn't happen. So we can do things in propagation to be able to overcome or to start different signals to the seed to be, it's fine, it's okay, it's time to grow. And so whether that's actually the physical hardness of the seed, and we'll break that down or scarcify it, or if it's, you know, like overcoming that a hormone specifically, or even just like some of the acids involved in the plant, or some of the chemistry. Environmental conditions are another ways that we overcome and tell the seed, this little baby inside the seed, that it's time to grow. And so sometimes, and the sad truth of life is that a lot of times the seed did not develop correctly. And so when we grow and we collect seed, especially like acorns or other types of seeds, you can float them. And this is just a real easy way. You guys can do this at home. And just, if the seed floats, then the embryo did not quite develop. And so all you can see, this is bladder pod here. And here we, all these types of seeds, you can see they're actually a little bit different color too, part of that undevelopedness, will float to the top. And so those aren't viable, most likely are not viable, and they won't germinate. But the seeds that sink, they have a nice developed embryo, everything's ready, and that's all packed and ready to go. Those are most likely going to be viable seeds, meaning that they are able and they have everything they need and they will grow most likely. So here's a little test here that you can use at home to test whether or not you have good seed. So again, when I was saying about that dormancy, there's a lot of things involved with the seed. There's a lot of signaling. Usually hormones are signaling type chemicals, and that all are involved with whether or not that seed is ready to start and start to grow and establish in its new environment. So there's a few methods in overcoming seed dormancy. And dormancy, we mean it's dormant. It's not living or growing. In fact, I think on this slide, what I was mainly trying to say is that the seeds are dormant, or it's actually suspended animation is another term for it. These are not actively growing. They're really slow. They are still using and living, and remember, it's a living little baby inside that seed. And it is still using nutrients and sugars and things like that, and the energy within that seed. It is still consuming energy to be able to live. So that's why you have what's called seed viability. So how long does seed last over time? Some seeds last a very long time. So Ceanothus, or the lilacs, that seed lasts so long if stored correctly, which we do here. But there's also seed that's very, very short-lived. A lot of your wetland riparian type plants are like that. And so there's some of those species, we literally, once it's in seed, we'll cut a branch and put it directly onto a seed flat that day, within that day. Acorns are another thing. Acorns, they drop. You got a short window to germinate those acorns. They don't store well, and animals are taking them. It's feeding. It's the keynote species that's taking care of an entire habitat. But the ones that do live and that aren't completely consumed by insects or things like that are the ones that will survive. So that's where viability and the dormancy of that seed is very important, that with how long they live and how you can overcome them and stimulate them to then grow. So we do it a couple different ways. So there's physical methods, and so sometimes if you have a real large kind of hard seed, you can actually take a file and break down and just get a little bit of that open, and then when you can then start to sow that seed and have a new radical root come out and then start to grow up from there. Sometimes it's taking cues from temperature, kind of mimicking what happens naturally in the wild. So there's a thing called cold stratification. So like physical stratification is like actually taking a file and breaking that down. Or there's cold stratification, which is actually a method in which you store it and hit it with and keep it in a real low temperature as if it was a winter. That seed, you know, it's not trying to grow, but it's taking the fluctuation of these different temperatures where it gets real cold and then it warms up. That's the signal to that seed, hey, spring's here. And not like us, we have more wet kind of winters here, but sometimes in other parts of the world, that's more of how those types of seeds grow. We do it with a couple plants, especially that go up towards, in higher elevations and plants from there, but typically you can do what's called a cold treatment or a cold strat. And you do it for about a month or so, and then you can sow those seeds and have them grow, and they will then stimulate that seed to now grow. Fire. This is the most exciting one for us to do here at Tree of Life, but there are fire followers. So we have plants from the chaparral and natural fire is a natural part of ecology, and there are plants that require kind of that stimulation for it to grow, and that's how the habitat itself even reinvigorates itself and starts anew and continues into the future. And so we have to literally set some seed flats on fire, and I'll show you the next slide where we do that. And then chemical. And so again, that's part of that hormone where we'll use something like a gibberellic acid, which is a type of hormone that stimulates seeds to overcome that dormancy. And so that signal, hey, to the seed, it's kind of a little cheat in some way to be like, hey, I know that inhibiting hormone you have in the seed to make sure you don't grow. Here's one that counteracts that, and then we'll start to stimulate that seed to grow. But also, kind of mainly that type of chemical thing we use, we'll actually use acids. And so a lot of these stimulating hormones are acids, but we actually use a sulfuric acid, and that's a way to break down that seed coat and make it easier for that little baby to then grow. And what that is doing is we're actually mimicking the stomach gut of plants or of animals. So when that fox eats that toyon berry, it gets a little bit of that stomach acid, and it breaks down the berry, and they're also part of that seed, and then can then sow those seeds, and then they'll start to grow. And so you can see here, we do this with lemonade berry. That's one that has a real kind of tough, hard seed. So you have to have kind of the right concentration. And so you can see that Idea, our seed mother here, who sows all of our seeds, she's using a sulfuric acid treatment. And sulfuric acid actually will also start to clean the seed too. So it'll remove some of that kind of those fungus and things that might inhibit or eat the seed. And then you're also cleaning up and softening up that seed coat for the seeds to then germinate. And then fire. So I wish I had a photo of the actual plant here, but coming into summer, the Rhamnia coulteri, the Matilaha poppy, this is one of the largest flowering plants in California. It's this big, white, beautiful, or fried egg plant because it's got this real yellow center ball on it. That plant, their seeds need fire stimulation. And so these are a lot of plants from the chaparral, like the Rhamnia. And so what we'll do is here's Idea, and we're sowing seeds onto the flats like we would normally, but then we will add a layer of pine needles on top. You could use leaf litter, pine works really well. And so we put a layer on it, and then you light it on fire, and then Idea is really good at this. Like she's a really good cook too, and she can tell how to keep the temperature just perfect over the seed so it doesn't get too hot or concentrate in one area, or make sure that it gets hot enough, and adding them enough fuel. And then you'll see that the seeds will then start to germinate after that. Now, there's probably more than just the physical aspect of the fire to stimulate it, but it's part of that. And it might be just a piece of the puzzle where that charate or that the charcoal and the ash afterwards could be also part of the chemical stimulant to the seed as well. So some people will use kind of liquid smoke. And sometimes that helps with seeds, but I think it's usually a combination, I think, does a lot better job. So have fun, set fire, but be safe. We had to learn, we actually put metal along these seed flats. That's something we had to learn because we started it and these are plastic seed flats, and yeah, but that works pretty well. And then ferns. This one's a little new for us. So I come from more of a science background and bringing that towards propagation. And that's kind of what this talk is really trying to show you. But we're starting, and we actually finally have out available in retail for sale. So this is one of my favorite ferns. And so this is Polypodium, or a California polyplody. And you'll see this out on some of the hikes all in and around here. It's kind of a winter type fern, winter spring, with a little bit more supplemental water in the garden, and it can kind of hang around all year, but really this is their best time a year. Kind of when it's like a little bit more moisture, humidity, a little cooler temperatures for these delicate kind of leaves to work. You can find a place in your garden that has a little microcosm that's a little bit nice and cool and a bit shadier, and this plant will do great. But this plant, so these don't have seeds. This plant, they're called older plants, but they evolved to have a very unique life cycle and a very unique kind of life history. So these won't have a hard seed coat or anything like that. These actually have a very unique strategy where they're actually two completely different types of plants in one during its life cycle. So we call that alternation of generation. Sorry to take you back to biology 101 in your high school. I'm just seeing some people having flashbacks and post-traumatic stress, but we'll try to make it as simple and harmless as possible. So this is really cool. So here's the life cycle of a fern. It's very fascinating. So what you're seeing right here of the fern and the leaves and everything like that, that's one type of plant. That's called a sporophyte. And so actually, this giant sycamore back here is the sporophyte of that plant. Now, what it happens is you'll see, and they're doing it now. I'm going to pass it around the backside of a leaf here. So you can see all the little kind of color, that kind of orangish red color underneath. So that's what you're seeing here. Those are the sorus, or sori, plural. But those are all the spores in there. But it's actually way more than that. Those are all encapsulated groupings of what's called sporangium. And so inside that, think of it like a little Pokeball or like, you know, a little Easter egg type thing. Inside all that is where all the spores are. And so once you break all that down, and once those release, it's even more and more microscopic. These little tiny spores. And then once those start to hit the right conditions and have the most moisture and everything, they're very vulnerable to fungus. It's a tiny little gamete. And then that will germinate and create this small little plant here called a gametophyte. That gametophyte, it's just a small little photosynthetic little plant. And it does have a little root called a rhizoid, and it will actually then have the male and female parts, much like a flower or anything like that, and then it actually will produce a sperm and an egg. And you can kind of see the two different color differences on those little dots. And then it has to self-fertilize or will fertilize itself or other type of gametophytes, and then from that will be the new plant. But it's actually the same plant, but that is how it exchanges its genetics, and then it will have this, or genetic shuffling, and then from that a sporophyte will then emerge, or the leaves, and the actual bigger part of the plant. And kind of what you know as a fern is what you see as a sporophyte. And that's actually occurring with higher plants, but all that, and where the seed and egg and the spores or the pollen and egg all happens within the plant with the help of pollinators and things like that, and then a seed forms. So it's similar but different. And then when they store that seed, that's kind of that emerging sporophyte. So it's a way that plants, this type of plant, will regrow without seed. But it's the same type of biology is kind of occurring, but in a very different fashion. So here it kind of looks more in the wild. My dog Jack and I went out to go try to collect some of this giant chain fern, or the woodwardia, and we found it kind of right in seed, and you can, or in spores, and you can see that they have these little packs of little seeds, and you see all the sorus there with the little pokeballs in there, and it had a little sheath protecting over it, and then we could then isolate the spores using screens, because again we want to clean it to make sure that there's not material there that could then harbor fungus and eat that little spore. We grow out these little, and you can see this is under a microscope. You can see these little tiny little gametophytes growing on, and this is just a little grain at the end of a pencil there. You can see there's tons of these little gametophytes. You actually can see a spore right in this corner here that germinated out and then to grow. It's using light and everything to create and nutrients to make that whole bigger structure. And then you can see we've been watering and misting them, and then that new emerging little sporophyte will grow out into this new plant that we have now for you. So again, what I said about the adoption fee and everything like that, or the form, I grew these under a microscope, and it took a couple of years, so be careful with them. But they're actually nice and hardy now, and it's actually a little easy to grow, so it's just getting them to that stage was a challenge. But understanding the science and everything is how we were able to do that. It's probably also why you don't see many of our native ferns out in the trade, but we're trying to overcome that. Back to cuttings. So this is an asexual way of growing plants, or taking clones. And this is Gloria. Gloria is our cutting mother. She is a machine. She just literally, my very first day here, I was working, and I think my bosses thought it'd be funny to be like, hey, work with Gloria and see how many cuttings you can do. You have all this experience. I'm sure you'll do great. I think I had like 2 or 3 different cutting flats done, and she was moving on to something like 23, or she was well above me. And my bosses come and give me a hard time. Like, I don't think he's going to hack it here. I'm like, it was fine, but she's just an amazing person. She does all of our cuttings. So she's taken anything that was a cutting or a cultivar. Gloria had her hands on that, taking that cutting and growing it. So what that is, like I mentioned previously, it's a genetically identical plant. So if there's plants we want the traits of, we'll take a cutting. So you can see here there's a couple different types of original of this seaside daisy, and there's 2 different types here, and you can see the flower is different. So if you want one or the other, and there's a little bit different growth habits in that, so we want to take a cutting or a division of that to make a genetically identical clone. Now, this solanum over here, or this really rare actual nightshade from the islands, does really well from cutting. So it doesn't have to be just the trait. There's some species that don't do that great from seed, but they do really well from cuttings. So a lot of our salvias and sages are like that. And so what we do is we'll take a cutting, we'll literally cut, but that's just a very incomplete plant, right? And it's going to slowly die if it doesn't get new roots and start to pull up water and be able to live. And so what we need to do is use a hormone, again, a signaling chemical, to stimulate roots to then grow. And so that's how we do it. And then once you do, you have now a complete plant with roots and shoots and everything it needs to pull up water and nutrients and capture sunlight and grow. So that's how you do cuttings. But you need to understand the parts of a plant to be able to take a cutting. So it doesn't just happen at any point. So under this, you're going to go right about where the node is, or right at that branching, and you go right slightly under. And right there is a lot of what's called meristems, or meristematic tissue. And that's where the actively growing new cells occurs. So if you put a hormone right there, it'll actually, as those cells are starting to regrow and multiply, doing meiosis and everything, or just starting to split off into new cells and grow, you have that signal to, hey, we want roots. And it'll be like, oh, yeah, absolutely. And starts to now put on roots right at that cutting and right at that wound where that meristematic cell is. And then you have some roots that will form there. But at every point, you have these, right at the node, you have little buds or axillary buds. You do have an apex or apical bud at the top, or your dominant leader. You want a good dominant leader for a lot of good structure of trees. But these axillary ones are the ones on the side. If you break off the top apical bud, it'll actually stimulate the rest of the axillary ones to grow. And so that's all it has to do with different concentrations of hormones. That's more towards horticulture, but we'll stick to the propagation side, where that little node, or those buds, is where you're going to have the shoots to grow. So you stimulated roots, and then from those shoots, you'll have the new plant. So you can see meristems here. There's a whole bunch of different types within a plant. You have kind of these apical or axillary ones on the shoot meristems. You can see here, you can see these areas where they're having a lot of cells start to divide. They actually can eradicate viruses of a plant physically, not using chemicals or anything like that, by taking the little point there where you're having really rapidly dividing cells. Sometimes they're dividing so rapidly, the virus within the plant hasn't been able to get to those new cells yet. So you can actually divide and isolate very, very small in a lab and completely eradicate that virus from that plant. That's a little bit advanced, but it is possible. So you also have these rapidly dividing cells here in the roots. So roots are growing out, right? So right at the tip of that is where you have a lot of that dividing cells as well. So that's a root meristem. So when we take in cuttings, right, you have a very vulnerable plant, almost like a seedling. And so water loss is a big part of that. It's kind of a race as soon as you take a cutting to be able to stimulate new roots to grow. But that plant is still doing all of its processes of using water and nutrients, and it gases out water and moisture as it uses and pulls it up to transport chemicals throughout the plant. So we have to put it in a nice, real moist environment so it doesn't use as much or need as much. And you can remove the leaf surface area. So again, it's not a whole bunch of these breathing parts of the plant, and then a lot more of the plant to need to sustain with water and everything. And so we put that in a greenhouse that has nice humidity and everything like that. But once that plant then evolves, now it's evolved or adapted into a completely different environment that's not out in your yard. And if you did quickly take that cutting out, it's likely that it'll be too tough, and that plant's not hardened up enough to be able to live. So we have to gradually acclimatize them and get them acclimated to a new environment. So we'll get less and less humidity, we'll put them out in an environment but with shade cloth, so it's able to then toughen up, build up some more cuticle layers around an outer tissue of the plant. And so that's called acclimatization. And so it's hardening off plants. So this is more of an advanced method that we grow. So this is a Oregon grape right here, or a Mahonia. I think the new name is actually Berberis. And so this is a really unique variety called Golden Abundance. Beautiful flowers, gets a nice blue berry on it. Excellent, excellent bird plant and habitat for a lot of different species. Great screen, but the flower on it is incredible. It's like explosions of these yellow flowers all around it. So we want to take a cutting of this. It doesn't do that great from cuttings. And it has these traits, and even that, it doesn't really do that well from seed. So what do? And so one way that you can do it, and they do this with a lot of tropical plants, is called air layering. So we'll actually wound the plant, expose those meristems. So there are meristems within the branch itself on the inside. That's how plants grow outwards, right? Those tree rings, those are the meristems as they grow. So you can wound it, but you want to make sure it's a nice clean knife and everything, so you're not inoculating with anything type of fungus and things that get inside that wound, just like if you got wounded. And then we'll wrap a moistened either sphagnum moss or any type of real kind of peat, and then you can add a little bit of root hormone in that wound, like you would take from a cutting. You wrap it up, make sure it's nice and moist, and you actually will have roots form on that branch into this little pocket. And then you can come back later when there's a nice established root, cut it, and you have basically a whole big cutting, a new plant like this big. And so that's called air layering. So it's up in the air, and it's a way of doing that type of propagation. So we do that here with this Mahonia. And Gloria, she does all these, and she calls them tamales. She's like, oh, I'll make the tamales on the cutting. So they look like she wraps them the same. By the way, she has the best tamales. Don't tell Mike. I love Hilda, his wife. Gloria's much better tamales. Anyway, this is going to be filmed. Sorry, Mike. Sorry, Hilda. But so we actually use it as a prop code too. We'll even say, hey, can you make us this many tamales? We'll come back, we'll cut them, and then actually root them into like a nice sized plant like this in this 2-gallon. So again, that was a way of overcoming things with propagation, or these challenges. So the seeds don't do well. We want the traits specifically. So we don't want seeds because you'll have that genetic variation, that we want these certain aspects and attributes to the plant. But a lot of times cuttings just don't do well. Anyways, this is one of the rarest trees in all of North America, or tall shrub tree. There's only 8 individuals left in the wild on Catalina Island. It's on the back side of Catalina. We have some other mountain mahoganies species, but this one is super rare. And it's only in one little gully, on Wild Boar Gully on the backside. One fire, that thing is gone. There's goats, other things on the island. It's just really, really threatened. And these individuals are getting older and older, and it's lost its ability to produce seed for that species. So there's a much more common species around of Cercocarpus. This is Cercocarpus traskii. There's Cercocarpus blanchii that's more widely distributed, and it's called hybrid swamping. So all these individuals are circling around these last 8 individuals, and it's wind pollinated, or I think there's actually small insect pollinating it, and it's just swamping it out, so all the seeds are hybrid. And there's no hybrid vigor there. It's actually, those seeds come up, and they're not genetically viable. A lot of times they don't even germinate. And you're just, every year, just no success, no reproduction, no anything. And these plants are getting older and older, and losing actually more viability to be able to produce good seed. And they don't do well from cuttings at all. So what can you do? You could try air layering. That was one thing I wanted to try out in the islands. I've been working with them, trying to see if we can do that, because it's a huge long trek to get out there too, different terrains. So that might be a way to do it. But the method I was proposing, and we're going to try, is called plant tissue culture. And so this is a method of growing plants in a laboratory setting. It's using tissue. You can take like a little bit of a leaf tissue or anything and grow certain plants, but that's only certain plants. And there's a lot of research, and we're at a place now where we can really start to use this technology. So that's what I learned when I got at University of Hawaii, and now I'm trying to bring that towards California. They've used it for a lot of species out there. We're trying to use it here now at Tree of Life for production. So what is it? So it's typically called in vitro culture. It is growing plants in vitro, which the term in vitro just means in glass, so in a test tube or anything. So the nuts and bolts of it, in fact, my textbook for learning how to do this was called Growing Plants in Test Tubes. Like, wow, that's right to the point. But that's exactly it. And so it has a nutrient, agar, so it's like a jello, that, but it has everything the plant needs to grow. So this plant has no roots, but it's growing just fine, because pulling up through its, or transporting water through its tissues, and all that nutrient and things it needs. It has it's completely sterile in the environment there. There's no competition from bacteria or fungus or anything like that, or insects, and it just has everything it needs to grow. Perfect conditions, perfect light and temperature, and it's just how you can really eliminate all these challenges to these propagules to be able to grow. But it's also a way to mass produce plants. So a lot of these GMO variety type plants out there, they'll use plant tissue culture to be able to have the traits in there that they genetically modified and massively grow out these plants. Orchids, a lot of orchids have, all this effort in growing this perfect, beautiful orchid flower. And so they will, but that seed is not viable. You're not going to get that trait. It's not really easy taking cuttings of orchids at all, so they use tissue culture to do that. That's a lot of what they were doing out in Hawaii as well when I learned this technique. But it's a method to do exponentially grow plants. I'll go over that in a second. But it's also a way to have nice, clean, disease-free plants because you're growing them in this perfect sterile environment. And also, what I'm trying to propose to more people of what they were doing out in Hawaii, try to do that more here in California and basically anywhere, is to also use this method to be able to store plants, to be able to have living plants available of these plants that lost their reproductive capability, and I can have thousands of them on a shelf, which thousands out there is susceptible to environmental changes, pests, disease, and it takes a lot of space and resources. I can just keep these kind of just nice and healthy and change their diaper every once in a while, or that nutrient agar on a shelf in the lab, and we can conserve and save these species as they move forward. So there's a lot of places that are starting to do that. So you need a nice culture room with grow lights and nice and climate controlled and very clean. So you're not going to be able to contaminate those cultures. So this is out in Hawaii. This is at the Lion Arboretum where they're doing a lot of that conservation work. And this is on ours. We have a little trailer back here where we do it. And so I have some Dendromecon up here and some Arctostaphylos we've all been trying. So the stages of it are very different. So you're going to take a source plant. So in this case, this was for my Dudley or on my masters. And then you have to clean and sterilize it. So use a sterile or a bleach or a chlorine type solution to get rid of all that bacteria and fungus of that little propagule or cutting. And this you have to figure out the right concentration for the type of tissue within that plant to not kill the plant, but kill everything else and have a nice clean individual. So that was part of what the Masters was figuring that out. Then you have to grow and establish them onto a media, and then you can start to add hormones and tell the plant, hey, I need new individuals or new shoots and things like that. And that's called multiplicating. And so I always use this example of trouble with tribbles to explain it because it's spot on.
There must be thousands of them.
Hundreds of thousands.
1,771,561. That's assuming one tribble, multiplying with an average litter of 10, producing a new generation every 12 hours over a period of 3 days.
And that's assuming that they got here 3 days ago.
And allowing for the amount of grain consumed and the volume of the storage compartment.
It's very logical, Spock. So it's actually pretty spot on of using tribbles. So say you have one tribble or one plant, and you're able to replicate that plant or get 10 new plants by stimulating it with that hormone. You'll see this whole mass of new plants and new shoots grow from that. You then have you can divide those up, and you have 10 new individuals. You just had one previously. And so sometimes in Hawaii, there was only one individual left in the wild. And this is how they were able to create new plants, or at least chances for that one individual to not die or be lost and extinct forever. So those 10, you do the same thing. You add a new, you put all those individuals onto that multiplying media, and now you have 10 new individuals from that. Now you have 100 new individuals. So within just 6 times of changing the diaper and dividing these one individual, those all can get you a factor 10. You can get, hypothetically, you can get to 1 million plants in just 6 replications. So for my master's work, it was right when it hit that the Dudley, or the succulent that live forever, were being poached out of the wild. And a lot of those types that were being poached were rare, very rare, only occurred on one island. And they almost completely wiped out Cedros Island down in Baja for this one type of Dudley on there that's very unique. And it's very possible it can go extinct in the wild soon. And so what this was all just a problem of supply and demand. There was no supply. They were all out in the wild. No one's growing them. We're trying, but we're only just a drop in the hat to the demands of those plants out in other areas in the horticultural trade. So there's a lot of challenges with hybridization and growing and from seed, and you can't really take cuttings up from that plant. So it's perfect. I was in grad school and I knew how to do this and had the capability here, and so we're applying that towards Dudleya to be able to just flood the market with these really rare and really unique and demand of certain species, to be able to just drop that price incentive under there and be able to conserve these plants. So why, if that plant's only worth, on the market so much, why would you spend all this time and effort and money to go across the world to go harvest out of the wild? So that's what we're trying to do. And that's where this type of method can be used for conservation, but also towards production, to create new individuals with propagation. So once you've multiplied these plants, then you can then transfer them and divide those, and then switch the type of media. Now there's no longer the shoot initiating hormones in that type of media, but now you have a root hormone, and that will then stimulate new type of roots, and now you have a complete plant that you can then acclimatize out into the greenhouse, and outside of that glass environment have a brand new plant. Again, that can be a real challenging step because you went from kind of the perfect conditions within that. So the next step would be into a greenhouse and then out into the hardened environment. So we did that here with a certain type of manzanita that we wanted to take clones of. Manzanitas do terrible from seed, and the cuttings can be difficult sometimes too. They do well, but certain species can be a challenge, and so we were trying to overcome that. All right, so thank you. That's what I got.